Hello, everyone, and welcome to today's webinar, Breaking Speed Barriers, Unveiling Biological Insights with Label-Free Chemical Imaging. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speaker for today's webinar, Dr. Volker Schweikhard, application scientist for advanced microscopy and also the product manager for coherent Raman scattering at the Leica Microsystems based in Mannheim, Germany. Dr. Schweikhard, you may now begin your presentation.
Welcome everybody to today's webinar on breaking speed barriers, unveiling biological insights with label-free chemical imaging. Today, I will have a webinar consisting of 4 parts, where in the first part, I will bring everyone on the same page by giving you a background on label-free chemical imaging, a new emerging modality that allows you to image targets that are largely inaccessible by traditional methods. And once we're all on the same page, I want to introduce to you the benefits of the new Leica Stellaris CRS microscope employing a brand new next generation laser source that delivers the following benefits for you. This new laser source and the new microscope will allow you to explore order of magnitude faster multiplex chemical imaging, and in turn allow you to follow complex biological processes in living samples at previously inaccessible timescales. You will also benefit from a strongly improved SRS image quality that will allow you to acquire statistically significant biochemical image data in less time. And you will be able to benefit from an enhanced spectral precision also that will really give you new opportunities for a very precise label-free spatial metabolic phenotyping. So we have various types of contrast mechanisms available in optical microscopy. A lot of you will be familiar with fluorescence microscopy, with which we can very exquisitely label highly specific molecular targets and get a very high sensitivity in terms of even being able to image individual molecules. Fluorescence microscopy has more to offer than just the intensity in the images. There's also a contrast associated with the lifetime of the fluorophore in its excited state that can be used to either color fluorophores based on their lifetime information, or also to introduce sensing capabilities for the local microenvironment of the fluorophores. We have additional nonlinear contrast mechanisms such as second harmonic generation or third harmonic generation that basically provide some biophysical properties on the sample. And as I will focus on today, there's yet another type of contrast mechanism, chemical contrast arising from the intrinsic molecular bonds that are present in the sample. And here at Leica, we believe that all of these different types of contrast mechanisms will really work very nicely synergistically together in the future to provide more information than just the sum of each individual contrast. So let me focus today on vibrational or chemical contrast. How do we actually achieve this label-free chemical contrast? And the technology that we are utilizing here is called coherent Raman scattering. And I want to dive in by just giving you an example of such a coherent Raman scattering image. So this is an image of a fairly highly zoomed in region of a brain tissue in which you can see the endogenous lipid molecules in that sample. And so you can see that the brightest structures here are actually tubular structures associated with the myelin sheath beads that are wrapping around the individual neurites in this tissue. You can also see darker regions. These are the cell bodies. And you can see the very dark regions in the center of the neuronal cell bodies. These are the neuronal nuclei. These are devoid in lipids because they occupied large parts with DNA and protein. That's why they appear as dark regions in this lipid contrast. Now, this sample has, in addition, been introduced with a fluorescent label. And so we can, in parallel to acquiring this coherent Raman image, we can actually take a 2-photon fluorescence image of that fluorescent label, and we get this very complementary contrast shown in the middle panel here. And by providing the overlay between those two contrasts, you can really see that those two contrast mechanisms provide perfectly orthogonal biochemical and molecular information here. And so we term this coherent Raman scattering an orthogonal optical contrast mechanism that can be used synergistically with fluorescence. So in this slide, I want to give you a very quick overview over the major benefits of this imaging modality and the types of applications that we can support with label-free chemical imaging. So broadly speaking, as I said already, you can now image targets that were inaccessible by a lot of other traditional methodologies before. And one of the first unique points of interest for the life sciences community was that with coherent Raman scattering, it's now possible to perform chemical imaging at the speed that is adapted to the timescales that are required for following the dynamics of biological processes. So you can follow the dynamics of the intracellular lipid droplets in such a small intestinal organoid, for example. But the technology has evolved a lot, and so we now view coherent Raman scattering as one pillar of a multimodal optical imaging approach where we can freely combine multiple different types of chemical contrasts together with additional contrast mechanisms such as second harmonic generation or even labeled fluorescence contrast. In this case of this bone tissue, the osteoblast cells were labeled with M-cherry, and this is visualized here through confocal fluorescence. And so you can see all these different contrast mechanisms provide synergistic information on a fairly complex sample, such as this bone tissue. We can do this type of imaging in a variety of samples ranging from cells, two-dimensional cell cultures, three-dimensional model systems, to tissue sections, and even intact small model organisms such as this intact unlabeled zebrafish, where we are visualizing here the individual cells of the retina inside the eye of this zebrafish. In terms of the research areas that we can now support, we have a completely new window at any type of metabolic information that you could be interested in. So we can support cancer research by providing new ways to visualize the cellular heterogeneity that is present in cancer. We can also visualize metabolic responses of such cellular systems to drug treatments. And of course, we can do this both in such spheroid model systems and in actual tissues. We can provide answers for pharmacology, such as where do my drugs go inside cells or inside tissues, by directly visualizing unique chemical bonds that are only present in the drug molecules. And we can provide a whole lot of additional medically relevant information, in this case, visualizing unlabeled amyloid beta plaques on top of the entire intact tissue. And the specificity here comes from slight variations between the vibrational frequencies in the pathological versus the healthy tissue structures. So overall, everything of this is done using coherent Raman scattering, and we really view coherent Raman scattering as a multimodal optical discovery modality, optical discovery approach. So let me give you a visualization of how these data sets are actually taken. We have access to different vibrational frequencies in the endogenous molecules in our sample by a laser tuning approach. So this means we are taking one image at one given vibrational frequency, Then we tune our laser, take another image at a different vibrational frequency, and so on. And we can build up an entire hyperspectral image data set in this way, such as represented here by a few representative images of such a data set from an untreated slice of apple, where on the right-hand side you can see the water content, and going to the left you can see different layers of the cuticle of the apple out to the outermost layer consisting of these very characteristic waxy components here. You can see the carbohydrates making up the cell walls, and you can see various pigments in this apple as well. We can now take the spectra that are present at each image pixel and visualize them. And you can see here that there's a highly detailed spectral fingerprint for each of these regions that we have available here. And you can also see these spectra are fairly complex, so they are somewhat convoluted, but we can definitely use them to analyze the biochemical composition of the material that we have at hand here. And we can also use those spectra as a basis to do what we call spectral unmixing of such a data set, where when I say spectral unmixing, I mean this. I mean a mathematical operation that allows us to cleanly separate the different biochemical constituents of our sample. So let me give you a state-of-the-art application example of what we can do with such label-free chemical data. This is label-free metabolic phenotyping in cancer cells. So it all begins with a spectral data set like the one that I've just shown. Here I'm visualizing it as a movie as we progress through this set of vibrational frequencies that we are probing. And you can see how those cells are lighting up in certain parts of this scan, but not in others. You can see how different little subcellular structures light up in different characteristic patterns as we go through those different vibrational frequencies. So you can sort of visualize the different pieces of information that are present here. But of course, this is a very complex data set to simply digest by looking at it. So what we typically do in such situations is we extract the characteristic spectra that are present in such an image by defining certain regions of interest, such as this region of aqueous buffer in between the cells, these subcellular lipid structures, the entire cell bodies, and you can see how each of these regions has a very characteristic spectrum. We can also compare these spectra then to separately taken reference spectra of pure compounds that we suspect to be present in this sample. And we can use then either the extracted spectra or such reference spectra as a basis for unmixing our data set. And the result is shown here on the right. So you have a perfectly clean unmixed image showing these unstained U2OS cells, showing the nucleic acid channel, protein channel, lipid structures, cholesterol deposits. And what is not shown in the composite image, but I'll show separately, is the water content. So I'll step through the individual channels. So nucleic acids localized nicely to the nuclei, protein present in the nuclei as well in forms of histones, but also, of course, distributed throughout the cytoplasm of the cells. Lipid structures much more localized in these point-like lipid droplets. The cholesterol deposits forming a very different subset of localized structures. And the water content, very interestingly, basically shows a negative image of the cellular structures because water is most prevalent outside the cells and is repressed more and more as we go through the cytoplasm and into those lipid structures. So very detailed biochemical information is here undoubtedly. And what we can now do is we can actually compare the extracted spectra from these different cellular regions to those reference spectra of the pure compounds, and we can do a very clean chemical composition analysis. So here, in this case, we have fitted the spectra of the cell bodies, and we have obtained fitting coefficients that are displayed here that give us the relative contributions of those different chemicals that are highlighted here. One note of caution, these are not directly concentration percentages because we haven't referenced those signals to those of pure substances, but in principle, such relative concentration information can be obtained. We can repeat this kind of analysis with the lipid droplets, showing here a breakdown into 4 possible different lipid subtypes, and it turns out 3 of them are actually found to be present in this lipid droplet. And we can analyze the cholesterol deposits here and really confirm that these are consisting of pure cholesterol. So this is the kind of detailed biochemical information that you can obtain from such a data set of unlabeled cells. Now I want to transition and give you a bit of a background on how we actually excite those molecular vibrations with light to obtain our chemically specific contrast. And the basic mechanism underlying it all is the spontaneous Raman effect. So in the spontaneous Raman effect, we have one pump laser exciting a sample or shining on a sample of molecules where the energy of the photons is actually not high enough to drive electronic transitions. And so we don't expect any fluorescence. So most of the light is actually transmitted through the sample. And very rarely will it happen that an inelastic scattering process causes one of the molecules to vibrate and causes a lower energy photon to be released, which we call the Stokes photon. And the reduction in energy of the Stokes photon is exactly equivalent to the to the energy of the molecular vibration that we excited. So that's how we get the chemically specific information transferred to the light. Now, these processes occur through so-called virtual states, meaning they are very, very rare. And then those spontaneous emissions leave our molecules vibrating. But such processes, like I said, are very rare. Only a tiny, tiny fraction of the photons are scattered in this way. So using this kind of an effect as a microscopy contrast is possible, but this kind of microscopy is very, very slow. And so researchers have been searching for ways to give this Raman effect a boost. And what came out of this is what we call coherent Raman scattering. So here we shine a second laser beam in addition to the pump laser beam onto our sample of molecules. And if we match the frequency difference just right between those two laser beams, such that it matches one of the vibrational frequencies of the molecules, then we can cause all of the molecules to vibrate very efficiently. And what we then have as a result is a loss of photons in the pump beam and a redistribution of photons into our lower energy Stokes beam. And we can actually visualize this again through such a diagram, an energy diagram, where now we are driving this transition down from the virtual state with that second laser. So it's a stimulated process, much in the same way as stimulated emission depletion, STED microscopy, or also stimulated emission taking place in lasers, making all of these processes much more efficient than the spontaneous variants of them. We drive those processes with picosecond laser pulses with fairly high peak intensities. And so we get, as a result, other nonlinear optical processes happening at the same time. And one of them is the so-called CARS effect, coherent anti-Stokes Raman scattering, where you have a cascade of photon absorptions and emissions resulting in a blue shifted photon that is fairly easy to detect. And so we turn these molecular effects into an imaging modalities by coupling the two laser beams into a laser scanning microscopes and detecting the appropriate effects. We're detecting changes in the intensities of the excitation light in stimulated Raman scattering and the emission of blue shifted photons in CARS. Stimulated Raman scattering, turns out, is a very quantitative, quantifiable imaging method that also has the full spectroscopic imaging content that you would have from spontaneous Raman scattering, whereas CARS is much more suited for a qualitative, rapid, label-free chemical analysis of very abundant species. But the common underlying mechanism behind both technologies is this vibrational resonance, the matching of frequencies between the lasers and the molecules that gives us this chemically specific multi-photon contrast. And multi-photon is a keyword here. Multi-photon microscopy is able to perform three-dimensional imaging because the signal is only generated inside the laser focus volume. And so we are able to translate the laser focus in 3 dimensions through our specimen, and in this way build up three-dimensional image information. The instrument with which we perform this kind of microscopy is called the Stellaris CRS, which is Leica's turnkey instrument for label-free chemical imaging. And so here I'm showing a portion of the beam path that is only related to the coherent Raman scattering. The blue box in the background here is the picosecond optical parametric oscillator. That is our light source for coherent Raman scattering, which emits two beams, one being tunable and the other one being fixed. And the tunable beam is actually used to address different chemical vibrations, again, with the appropriate frequency difference between the two laser beams. And this picosecond OPO light source is actually the reason why we are here today, because we are about to present to you a next generation light source that has much enhanced properties compared to the current generation. In terms of the microscope, you have here a fully integrated Stellaris visible confocal microscope with additional modalities added on top, CARS and SRS, as I explained in the previous slide. In addition, we can take second harmonic generation signals and 2-photon fluorescence signals of the same sample. So with this slide, I want to show you how integrated this coherent Raman technology is into the actual user interface of our confocal microscope. I'm going to play a little video of how we set up such a coherent Raman scattering experiment. We basically have to activate the two laser beams, the tunable and the fixed laser beam, where we can access here the Raman spectroscopic information or the wavelength information. We can control the intensity of the laser. We have to open the laser safety shutter and then activate the SRS detector or stimulated Raman detector. And then we can start to capture image our first vibrational image. And so what you can see here is such a brain tissue as I have introduced it to you already before. So this is how easy it is. You basically operate an instrument where the user interface is very much akin to that of a confocal fluorescence microscope. So now let's come to the benefits of the new generation of the Leica Stellaris CRS microscope. And let's start with the first and most obvious one. The new laser source allows us to explore an order of magnitude faster multiplex chemical imaging than in current generation instruments. And so I want to give you, first of all, an introduction to some of those game-changing specifications of this new laser source. You can see here a comparison of the current generation Pico Emerald S laser to the new generation Pico Emerald FT laser source, where in terms of being able to tune from one user-chosen vibrational frequency to another user-chosen vibrational frequency, it used to take about a minute or even longer to perform that highly precise tuning step. And the laser manufacturer has now managed to reduce that time to just about 1.5 seconds. So really a complete, game-changing change in terms of time scale. Such vibrational scans were already implemented in a fairly efficient tuning fashion with about 6 seconds between images previously, and they were able to reduce that even further to about 1 second now. There are additional beneficial features through a redesign of the optics in this laser source that give this new laser source a signal-to-noise improvement for stimulated Raman scattering on the order of 10 times. And part of this has been achieved by a reduction of the repetition rate of the laser pulse train from 80 to 40 megahertz. But this is a reduction that is not of consequence in almost any of the applications that I'm going to present today. So this is not just a numbers game. Those improved tuning timescales now allow us to access and enable groundbreaking new applications in the life sciences. And so I'm going to first show you what it actually means to now be able to inspect a sample based on its chemical contrast on the fly. So going to play a little video here. We are actually scanning a sample of cells live as we go. And I'm changing the vibrational frequency here now to go from the lipid structures to the water content. And you can see how within a second or a few seconds you have that change of contrast. Now we're switching it to the, the protein contrast in this sample. So a very different type of contrast. Now I'm changing it to the unsaturated lipid bonds. There's not much of an unsaturated lipid population in the sample. That's why it's a fairly dark contrast. Now changing back to the total lipid content for a change. So you see, again, all the lipid structures. And then lastly, we're changing to a very specific lipid, namely cholesterol. And you can see cholesterol now coming up in just a few very specific locations inside the sample. And so this is a type of exploration that is now really at your fingertips, and you would have to wait. In the previous generation of the instrument, you would have had to wait about a minute for each one of those contrasts to show up. So it's an entire new way of thinking of how to explore the chemical information content in your specimens. Now, how can we turn that into a controlled experiment? So I'm going to show you here now a very breathtaking example of multiplex chemical imaging in a living organism. So I'm going to play the video without much further ado. So this is a late-stage Drosophila embryo, a live organism, where we are switching continuously between imaging the protein content and the lipid content in this specimen, while at the same time in parallel acquiring this blue additional contrast arising from second harmonic and two-photon fluorescence, showing yet a different kind of structure inside the organism. Now, the time scale here is about 16 seconds per time step. So within 16 seconds, we take those two images and perform two of those tuning steps. And you can see this movie lasts about 440 steps with a total runtime of 2 hours and in excess of 2 hours. And you can really follow the dynamics of both the protein and lipid constituents of this specimen without, as you can see, doing any harm to the organism. We can break this down into the individual channels, but the assignments here are somewhat tentative because it's really hard to actually assign specific organs to a developing organism. But you can see there are definitely more protein-rich organelles that you're looking at here, or cellular structures, and also more lipid-rich ones. And then you have this additional biophysical contrast mechanism arising from second harmonic generation. We can push this fast multiplex imaging even further. So here I'm showing you an image at an 8-second repetition rate where we were able to track cargo transport in plant roots. So you can see here an overlay and the individual channels showing the carbohydrate content and the lipid content or content of waxy compounds in this plant root. And in the overlay, you can very nicely see that even for those fast-moving vessels, we have very, very limited amount of motion artifacts. You can always see the two colors being almost perfectly on top of each other. So we can track multi-component cargo transport, even in such very fast-moving organelles, such as the ones in those plant roots. We can use those capabilities also to do volumetric multiplex chemical imaging now. So this is a Z-stack where in each image plane of the Z-stack, we are tuning between the carbohydrate and the lipid content to have the two images in each plane tightly registered to one another. And we can do that with about 2 minutes, 234 seconds per time stack. And you can see that time resolution is fast enough to follow the dynamics of such a growing plant root. We can even do things that were considered totally crazy before. We can do spectroscopic imaging of a living specimen. So you can see here, we're doing what was called a spectral scan of the high wave number region, where we are tuning from the right through the water contrast, through unsaturated lipids, through the carbohydrates, and then to the lipid content. And you can see we can get very meaningful spectra of individual such vessels and also an individual portion of such a cell wall. And we can do this kind of spectroscopic imaging now in 2 minutes. So yes, we do have some motion artifacts here, but this kind of a spectroscopy would have been virtually impossible with previous generation instruments. And the spectroscopic information we get is real. these spectra resemble those of oleic acid for the lipid components and of pectin, which is a type of carbohydrate. But we don't see much evidence for cellulose here. So this is highly actionable information that we get. Okay, so I told you there's more benefits than just the faster tuning. We also have an improved stimulated Raman scattering image quality. So let me show you that. And I hope the broadcast transmits the fine features of those time-lapse movies. So these are time-lapse movies of, excuse me. These are time-lapse movies of a stationary sample of polystyrene beads. And so within the individual beads, you can see the noise on the left-hand side with the old laser source compared to the noise for an identical sample of beads excited with the same kind of excitation power. And you can hopefully see the stark difference in the signal and noise in those two images. So now you can ask, okay, I'm happy with this enhanced signal to noise, but what can I do with it? You can, for example, ask how much faster could I go with the new light source to achieve the same signal to noise as with the old light source. And it turns out that you can go about 8 times faster. So you can do 8 times faster single color chemical imaging with the new light source than it was previously possible for the same signal to noise ratio. Or you could ask, I'm happy to trade some of the signal to noise signal contrast that I have to reduce the laser power to do gentler imaging. And so here's an example of this. This is actually such a Drosophila larvae after it has hatched. We've immobilized it in agarose. And so you can see here 2 time-lapse movies of the head region and of the tail region of the same animal, actually. So these were time-lapse movies taken back to back. And you can see how this animal is freely behaving and not changing its behavior over the time course of about 1,000 frames on the head region and 700 frames on the tail region. So obviously a very gentle kind of imaging that we didn't perturb this animal in its activities. And with this, I want to come to the last major application area, how we use the enhanced properties of this new laser source, namely the enhanced spectral quality that we can achieve. And we use this to do a more precise label-free single cell metabolic phenotyping than was previously possible. And I've shown the principle already in the beginning. So we take those spectral data sets, and I'm not going to go through the details of how we generate masks here. I want to say we can now basically confine the spectral information either to the cell bodies or to the lipid droplets to basically extract specific spectral information from only parts of the cells. And then we can compare that spectral information again to reference spectra of pure compounds. We can now actually appreciate how good the spectral data of such an image of this new light source is by comparing the measured spectra to a model fit consisting of just 5 major biochemical constituents that we expect to be present in those cells, and we can achieve this really amazing concordance between the measured data and the fit. We can do the same kind of analysis on the lipid droplets, and you can see here we pull 3 different types of lipids out, and of course, very different chemical composition than the rest of the cell bodies. And so this is more or less what I've shown you already at the beginning. But now we are going to repeat this kind of an analysis for cells undergoing or having undergone different metabolic perturbations. So we can supplement such cells with palmitic acid, and you can see already visually that there are more lipid droplets present in this sample now. And if we do a chemical analysis of those lipid droplets, you can visually appreciate how different the spectra of the lipid droplets are, and that is reflected in a different chemical composition where we now indeed have palmitic acid as the major constituent of those lipid droplets. We can do a similar type of experiment with oleic acid, and oleic acid was already the major constituent of the lipid droplets in the native state. But with the supplementation, we are able to push even more oleic acid into the lipid droplets. We can repeat it with linoleic acid. This is a doubly unsaturated fatty acid, and we can again analyze the spectra, confirm that the major constituent is now linoleic acid. We can repeat the experiment with cholesterol. Again, very different spectral shape, at least the fine details are very different. You can also see the localization behavior of cholesterol in those cells is different. It's not so much localized in lipid droplets, but more in such larger deposits. And if I may speculate, cholesterol is known to be associated with cardiovascular disease, and you would find cholesterol in atherosclerotic plaques. And so we may speculate that these deposits of cholesterol that we see here are actually somewhat of a cell culture equivalent of such pathological structures. We can do additional types of perturbations like deplete lipids, remove cholesterol specifically, or deplete the cells of glucose, and we can analyze the responses of the cells. Under each of those conditions, we can pull out biologically meaningful changes compared to the native state. And so the real significance of this approach is really shown by a recent landmark publication where this very similar approach, extracting a sparse data set of metabolic markers, key metabolic markers from such a spectral SRS data set, in this case, proteins, carbohydrates, fatty acids, cholesterol, and nucleic acids, performing this kind of analysis on a cancer cell line and then subjecting those cells to chemotherapy treatment. And in the single cell analysis of protein content, carbohydrate content, fatty acids content, and cholesterol content, you can see statistically significant changes as a response of the cells to the chemotherapeutic treatment. So we can really see a metabolic response of the cells to the treatment. The same experiment was repeated in a resistant cell line, and it was confirmed that there were no statistically significant changes in this metabolic panel. So you can really see highly specific metabolic changes of responsive cells versus non-responsive cells to treatment. So this is highly significant, of course, for drug development. You know, have a metabolic readout to see if your drugs are having an effect on your specimen. Okay. So with that, I'm going to more quickly walk through a couple of applications of coherent Raman scattering that should give you more or less just an idea of what you can do with this type of capability. So I've already shown this image of a cancer model system where the real unexpected finding was that of an unexpected rare phenotype. So this is a phenotype of a sparse subset of cells in the core of this spheroid structure that is very rich in lipids, but almost all exclusively saturated lipids, so a very different spectral profile than in the rest of the cells. So that's a very unexpected finding that we still haven't had the time to explore more. What we can do in this kind of an setting is we can explore the capacity of our approach for phenotypic discrimination of the different cell types. And so here I'm showing the spectra of individual lipid droplets from the different regions here, and you can see how much of a difference in terms of a large number of spectral lines is present here. I don't have the time to go through all of them in detail, but we think that within the lipid component of cellular or tissue samples, there should be plenty of information content to allow for a cell type discrimination or even phenotype discrimination. And we can, of course, use similar approaches to check the response of such spheroids to chemotherapeutic treatments. We can, of course, look at different organoid samples. This is a very fascinating one, a pancreatic organoid, where we have, again, the cellular and subcellular organization in the three-dimensional specimen. And here, one of the fascinating findings was this very prevalent occurrence of these very bright lipid and protein-rich vesicles that we think are precursors to insulin secretory granules. So very fascinating research questions opening up through such images. We can perform multimodal imaging. I've already highlighted this example, so I won't go through the details too much more, but just highlighting once more how we can seamlessly combine fluorescent information with various bits and pieces of biochemical information and biophysical information to get a fairly good idea of actually the bone formation process in such a sample of a skull cap of a developing mouse. And of course, our customers are now using this type of approach to also study diseases of bone formation or bone degradation. So again, highly medically relevant information present here. And again, relies on this full integration of all the different modalities in this instrument that we have at our fingertips now. We can, of course, do this imaging also in very tiny samples. Bacteria here shown just for, proof of principle. We can even look at individual lipid nanoparticles that are often used now for drug delivery. We have the sensitivity to look at such individual particles. We can do this kind of imaging in very large samples, such as this full mouse brain slice here, where we can then focus on regions of interest, such as this cortical region, this one of a mouse grown on a regular diet. We can compare it to the same region of a mouse grown on a high-fat diet. We can analyze the differences, and they are in part obvious, but of course, in part, they would have to be dissected in more detail by looking at the spectroscopic information. We can look at organoid or spheroid model systems. So this one is a very fascinating one. Osteoblast spheroids are shown on the top here that were brought in contact with a spheroid of cancer cells, and where we can see basically the migration of cancer cells onto the osteoblast spheroid. And so in this way, you might have a model for a cancer cell migration in 3 dimensions. Very fascinating to look at. And of course, we can do this all in full 3D. A last point that I want to highlight is everything I've shown you so far was focused on this label-free aspect, but of course, nobody prevents us from putting labels on molecules as well, which is what we call vibrational tags. And those vibrational tags are much more than a way of visualizing small molecules. We can actually produce metabolic activity information in this way. We can feed animals unique molecules such as deuterated glucose or deuterated water, and then watch the metabolic incorporation of those deuterated bonds into newly synthesized proteins, newly synthesized lipids, and other newly synthesized biomolecules. And by doing a spectral analysis on those newly synthesized molecules, we can find different localization behaviors, of course, different time points in the development of an organism, different responses to disease conditions where those unique bonds are being incorporated. So very, very fascinating information coming from this kind of an approach. These Raman tags can be used as activatable probes. So if you couple an enzymatic activity to the generation of unique molecular bonds or the destruction of unique molecular bonds, then you can turn signals on and off depending on enzyme activities. You can also modify the chemical environment of such a chemical bond, and in this way tune the frequency of the vibration. And in this way, you can generate an entire palette of unique, spectrally distinguishable vibrational signals that you could now conjugate to a different set of each one of to a different biomolecule, and you have a new way of doing optical multiplex imaging with vibrational tags. So with this, I want to end up with a teaser. We can also combine single photon arrival time information with, for example, cars information. And so here we have both the intensity of a car's image and the arrival time information in such a car's image. You can see the blue color signifying highly instantaneous arrival time. So basically photons, excitation photons arriving with, cars photons arriving instantaneously with the excitation by the laser, which of course makes sense because cars is an instantaneous process. But we can add glucose to those cells, and then we can see those lipid droplet structures disperse, and then some more longer-lived fluorescence type information becomes available. So very fascinating combinations of chemical and fluorescence contrast will become possible in the future. So with this, I would like to come to the end of the webinar by presenting you the rapidly growing diverse range of applications that we can now support with label-free chemical imaging. We have basically a new window at various disease conditions, oncology, neurodegenerative diseases, basically any disease with a metabolic signature. We will have something to say with label-free chemical imaging. So we can support fundamental discovery research towards advanced pathology applications, basic tissue analyses, spatial omics with a focus on metabolomics. We can support drug development for pharma, where we can basically watch the drug delivery to cells and tissues by looking at the molecules themselves, penetrating through skin as well, but also by looking at the metabolic responses of the cells and tissues to the drug treatment. So this gets us to mode of action questions. We can support cell and tissue biology, any question that involves metabolic dynamics, for example, in stem cells, developmental biology, any application requiring a chemical phenotyping. Neuroscience, there's lots of work going on already now in terms of brain development, brain metabolism, and neuroimmunology, neurodegenerative diseases I mentioned already. A lot of interesting applications coming up in metabolic engineering in terms of bioengineering research, but also tissue engineering. Model organisms, I showed some examples. Basically, the development of these small model organisms is very amenable to label-free chemical imaging. Material science, very fascinating topic, has not been one of the early adapters of this technology, but we think that applications in polymers, microplastics are coming up very strongly, connections with food chemistry as well, cosmetics, so lots of application areas in this space as well. And then I want to highlight it one more time, vibrational tags, a very fascinating and active area of scientific development at the moment, where we are now able to watch metabolic dynamics in ways that we haven't been able to look at in vivo in any other way before. These applications are also covered in a large body of literature that is already existing using coherent Raman scattering. I don't want to go through those details. We have a publication list up on our science lab that I will show up later. But as your key take-home message, you should think of spatial metabolic phenotyping as a new way of looking things in disease, in development, in aging, supporting drug discovery and supporting fundamental discovery research in cell and tissue biology. So like I said, we have resources. If you are curious for more, the Leica Science Lab has a collection of articles from the practical ones, how to prepare samples, but also to the more application-centric ones that I just showed previously. And if you're curious to see some of those publications that I just highlighted, we have an up-to-date publication list on Leica Science Lab. Feel free to reach out to me if you can't find the link. And of course, we have a new product page for the Stellaris CRS with the new Pico Emerald FT laser source for you to look at some of those applications with a little bit more time to check them out. And so with that, I'm ready to take your questions. Thank you very much for your attention.
Thank you for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's get started here. Our first question says, hello, I was wondering how specific you can get with the identification based on the spectrum analysis. Could you identify specific proteins or class of proteins?
Yes, thank you very much for this really very good question. And I have already posted links to two publications into the chat. I hope you can see them. These are publications giving reviews of Raman spectra of proteins and Raman spectra of lipids. And so if you browse through the data that are shown there, you can get an idea of the spectral differences that you can have available with different types of proteins or different types of lipids. Now, in general, if you are talking about proteins are mixtures of 20 amino acids. They are the typical average composition of these proteins is very similar. And so you will also get very similar Raman spectra. Now, that being said, there are subtle differences. So if you had, for example, a pure collagen protein sample, you would likely be able to distinguish it from many other different type of protein samples. Now, if you're talking about a mixture of proteins like you have in biological samples, like in biological tissues or cells, it will become much harder because you have so many different types of proteins that all have very similar Raman spectra. So it will be very hard to pick out one particular type of protein on top of the background of all of these other biological proteins in a tissue sample, for example. With lipids, the situation is a little bit more favorable. It turns out that there are several lipid subclasses that we can easily distinguish. We can easily distinguish fully saturated versus unsaturated fatty acids. We can also distinguish esterified versus non-esterified fatty acids. Cholesterol has a fairly unique spectral signature. So there's many different lipid subclasses that we can resolve, distinguish, different other types of small molecules, carbohydrates, nucleic acids, that we can distinguish. But again, as for going for an individual class of proteins within a biological sample, that is going to be very hard and only in exceptional cases possible. One possible case that I have shown already is aggregated protein structures where you have spectral differences based on beta sheet versus alpha helix content. So there are some specific cases where you can make distinctions between protein classes. I saw that there were some related questions in the chat. For example, the one question was how fixation of the sample will modify the spectra, and if it is possible to even work or make such an analysis on fixed cells. And this example that I showed of the spectral phenotyping was actually done on fixed cells because they have the benefit of not moving during the spectral analysis. So you will get actually much cleaner spectra from fixed cells than from live moving cells. That being said, of course, the fixation process may have an influence on the chemical composition and on thereby also the Raman spectra. It turns out that formalin fixation or PFA fixation is fairly harmless in terms of changing the Raman spectra. So that is the preferred way of fixing cells for Raman studies. What is discouraged is methanol fixation because methanol fixation tends to wash out a lot of the endogenous biochemistry of your sample already. So formalin fixation definitely preferred. And another question in terms of specificity was if it is possible to distinguish cellular structures such as centrosomes or microtubules among intracellular proteins. And there I have to say, I have not yet seen a publication showing that kind of specificity. But since this is all still a very much developing technology, the kind of spectral quality that we have available now, combined with the spatial resolution that we have available now, might enable application like that in the future, but it's certainly not going to be an easy one. So this might be worth an exploration. So I think that pretty much covers that complex of questions.
Excellent. Thank you so much. All right. I moving on here, our next question is asking, is the process able to resolve one cell from its neighbor cells, meaning we visualize cell junctions using the system?
Yes, another great question. Also in terms of, again, referring to specificity and spatial resolution. So you may remember the intestinal organoid that I showed. And there I just used one contrast that showed a combination of lipids and proteins. And you might have been able to resolve with your eye individual cells there. What I can say in general is you will see a decrease in the protein content towards the cellular membrane, and you will see signatures of the lipid bilayer, the cellular membrane. But these are not very strong signals because we're talking about a single bilayer of molecules here. But I would say in a nicely behaved tissue, like an epithelial tissue or an epithelial monolayer of cells, you will certainly be able to recognize individual cells. It becomes a little bit more difficult if you have many different classes of cells and cells sort of forming more difficult three-dimensional structures. But in principle, it is possible to resolve cell junctions. Yes.
Okay, fantastic. Thank you. All right, our next question here is saying, you mentioned a guide on sample preparation. What are the most important steps?
Yes, good question as well. In principle, you want to have as little processing steps as possible because you want to preserve the endogenous biochemistry. So ideal samples are fresh samples, live samples, or fresh tissues that have, let's say, only been formalin fixed. So that's the ideal preparation method. But in addition to just fixing the tissue, you also need to bring it into a sample format that is amenable to imaging. So stimulated Raman scattering is a transmission type imaging. So the detection is done on the transmitted light. So what we need is a sample geometry that has the sample sit on a 170 micron typical cover slip, and then sandwiched typically with a microscope slide. I think these are the most common, prevalent preparation steps, but you can, of course, work with a lot of different methodologies. And the general advice is to try to keep your biochemistry as unperturbed as possible. In terms of embedding media, I should mention that you want to use ideally something that doesn't have a strong Raman signature by itself. So water is a pretty good one. If you have access, deuterated water is even better because all the chemical bonds are shifted into a different Raman spectral region. But just plain water will work as well. Agarose will be a fairly good one, but you can also use glycerol-based mounting media like Prolonged Diamond or all the commercial ones available. You just have to be aware that you will have a Raman signature of the embedding medium as well that you need to consider. Yeah, thank you.
Excellent, thank you. Okay, next question here. Can the method recognize post-translational modifications of proteins such as acylation, phosphorylation, succinylation, etc.?
Right. Good question as well. So I am not aware of studies proving that, but in principle, all these chemical groups that you add to a protein have unique chemical or Raman signatures. The only limitations that you will run into here is you need to have a sufficient concentration of these post-translational modifications. And the spectral signature of your modification needs to be sufficiently different from the spectral signatures of all of the other biomolecules that you have around in your sample. So again, a very good question. I don't have a definite answer on that one. Certainly something I'm sure it will be tried in the future, and the answer will be available in a couple of years from now. Yes.
Excellent, thank you. And we have time for just a couple more questions. So this next one here is asking, is the Stellaris CRS able to be upgraded from existing Leica CARS or SRS system?
So that's a very difficult, and it depends on many configuration details. I would say, since it seems like you are a person who is already in possession of a Leica instrument, I would say talk to your sales rep. We can give you specific information on that. So some systems are upgradable, but others aren't.
Excellent, thank you. And for our last question that we have time for today, this question is asking, when do you think the method will be approved for IVD application for pathology?
Whoa, that's a very good one. So I know proof of principle publications are out there, or at least I'm not familiar with the specifics of that publication. So I know research is ongoing in those directions. But of course, IVD is one of the most sensitive places where you want to apply any technique. You need to be sure that you're not generating any damage with the light that you are subjecting yourselves to. So unfortunately, I don't have a good answer for that question. But I know, again, it's not out of the realm of possibility.
Wonderful. Thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Bye-bye.